So this is a research talk, but it's also going to be very, very much focused on the idea of this being a workshop where you can learn something new. So I think, although this is research, it's something that many of you will be able to use in your own work. And I'm going to present the work in a way that I think you'll be able to learn something about the biophysics of reactions at membranes that will be generally useful to you, even beyond your immediate interests. This work is by Kelvin Peterson and Boris Lipchenko and myself. It's available in bioRxiv. It's currently under review, and hopefully you'll be able to see the final version soon. But if you want to read about this in more detail, you can look up my name, and it's the most recent of my papers that's in the bioRxiv repository. So the overall problem is how to model membrane-associated reactions at the cell scale. And the reason that this is a problem is because of this issue of parameters. So reaction kinetics are commonly determined in, in vitro assays, or maybe even in cell-based assays, but using volumetric approaches, where you have a large number of molecules and you know what the volume is, and you know what the number of molecules is, and or the concentration, and you determine what the rate constants are based on these volumetric measurements. But the important thing to realize is that those measurements may not really be relevant to membranes. Membranes, you can think of the reactions at a membrane as actually being volumetric. Usually the reaction occurs between two tethered sites where the tethers anchor the molecule to the membrane, but the reaction occurs in the cytosolic region right adjacent to the membrane, or the extracellular region. There are very few reactions that occur within the lipid bilayer. Most of them occur at the interface between the cytoplasm and the membrane, but really in the cytoplasm, where the constraint is that the two reactants are tethered by various linkers to the membrane. So we have to worry about how to translate a measurement that might involve a volumetric measurement to this tethered membrane reaction, where the binding reactions may be identical, but now how do we account for this tethering that forces the reaction to occur on the surface? So that's the overall problem that we're tackling. And really, our motivation for working on this problem has been many, many workshops like this over the years, where this problem has come up over and over again. And now I think we finally have a way. It's a little bit complex, but we finally have a way to address it. Okay, so let me give you a little background. The idea of membrane reactions goes back, I guess, almost 60 years to this paper. It's a very famous paper, very highly cited, by Adam and Delbruck. Delbruck was a pioneer of biophysics. He was trained as a classical physicist in quantum physics, and then transitioned to molecular biology and became one of the fathers of modern molecular biology, and actually won a Nobel Prize for his contributions to that field, not his contributions to physics. But he published this paper with, I guess, his student Adam, called Reduction of Dimensionality in Biological Diffusion Processes, where he compared reactions in the cytosol to reactions in the membrane. And there's this cute quote here at the beginning saying a drunkard who's staggering around is wondering if he'll ever find home again. And this mathematician named Polya says, you can't miss, just keep going and stay out of 3D. So when you're a drunk trying to find home, you're tethered to the land, you're tethered to the surface. And according to Polya's theorem, mathematical theorem in 1921, a random walk on a surface is guaranteed to find its point of origin. So you can set off a random walk and it can keep on going in all sorts of directions and span a large space. But if you wait long enough, there's a probability of 1 that that random walker will get back to where he started, or get back to any other particular point on the surface that you want to designate. On the other hand, in 3D, that's not the case. In 3D, there's only a probability of about 0.3 that the random walker will ever get back to where he started. So there's another mathematician who just had another cute way of saying the same thing. So Adam and Delbruck recognized this, knew about this, and they thought more deeply about what it means for membrane reactions. And they translated this idea to biophysics, to reactions on membranes. And they said with a stationary membrane-bound target, like a receptor, a 2D diffuser can't miss, but it can miss in 3D. So therefore, diffusion-limited rates will always be faster on membranes. And therefore, it's advantageous for a cytosolic ligand to be absorbed onto a membrane before finding its receptor. So this paper in 1968, which was actually published as a book chapter, set off a lot of people to thinking about the importance of membrane reactions. But one of the key points here is that these are diffusion-limited rates. That is that diffusion is slower than the reaction probability when these two molecules meet, and therefore diffusion is limiting the speed at which the reaction can occur. So there were a series of papers that were based on this original work, but then more detailed thinking about and knowledge of membrane biology created some new ideas. And so there was a paper by McCluskey and Poo in 1986 that basically recognized that diffusion in membranes is really slow. Diffusion in membranes is a couple of orders of magnitude slower than diffusion in the cytosol. And after all, the cytosol isn't an infinitely large space. It's a 3D compartment, but it's confined. And so, maybe this idea of Adam and Delbruck isn't even that relevant. And the other thing to realize is, as I said, the Adam and Delbruck idea only works for diffusion-limited reactions, where reaction rates, the kinetics of the actual combination is faster than the diffusion. So only for diffusion-limited reactions. So another series of papers were published, one in 1984 and one in 2011, that recognized that the reactions on the membrane really don't occur on a surface. They really occur in a volume, as I mentioned at the beginning, where the reactant sites, if you want to think in spring solid terms, the sites that are reacting are just tethered to the membrane, but they can experience a certain volume below the membrane in the cytosol where they can react. So these two groups defined a concept called a confinement length. And you can think of that as the physical length of the binding site from the membrane, the distance of the binding site from the membrane. And if you have that distance and you have the membrane surface, you now have a 3D volume. The surface defined by the surface of the membrane and this confinement length just above the membrane or below the membrane, depending on whether it's extracellular or intracellular and what your convention is. So you really can think about this as a volume. And if you take the volumetric rate constant that, as I said, is sort of easy to measure, you can convert it to a 2D rate constant by dividing the rate constant by this confinement length. That just changes the 3D rate constant to a 2D rate constant. It takes out that confinement length. And so you can go from units of per micromolar per second, which is the unit of a 2D bimolecular rate constant, to units of per molecules per micrometer squared per second, which is the unit of a surface binding rate, just by dividing by H. Now, we'll talk about that a little bit more later. And then just a few years ago, there was a nice paper published in Biophysical Journal, which defines the concept of reach. And this recognizes that diffusion of the membrane protein cytosolic domain is confined to 3D diffusion and is likely to be much faster than the 2D membrane diffusion. So I mentioned before that diffusion, lateral diffusion in the membrane is maybe 2 orders of magnitude at least slower than the diffusion of a comparable molecule in the cytosol, very much slower. However, the diffusion of a molecule that's linked, or a site rather, that's linked to the membrane by a tether could be much faster. It could be, if the linker is flexible, it could be the same as the diffusion of the free molecule in the cytosol, because it is in the cytosol after all, right? If anybody has any questions, I'm willing to take questions in the middle. This is long enough that I can be interrupted. So this concept of reach was formulated sort of as follows. So let's take a specific example. Let's say you have a phosphatase that's a membrane-linked phosphatase. So this is the membrane portion, and the phosphatase portion is linked by a flexible tether. This is based on a real molecule, PD1, linked by a flexible tether, and can swing around and sort of occupy any area within this sort of hemispherical region, this volume, and find a phosphorylated substrate that it can dephosphorylate. So the idea in the 2019 paper is that this is fast, whereas membrane diffusion is slow. So if membrane diffusion is slow enough, the the longer this reach is, the more likely it is, the larger this L, the length of the tether between the phosphatase anchor and the active site of the phosphatase, the longer that tether is, the more likely it is to find a phosphatase. Very simple idea. The more likely it is to find its substrate. And they did some simulations and they show that that's in fact true. However, in the domain where membrane diffusion, where this thing is starting to move more quickly, this turns out not to be true anymore. And in that domain, and that's why these curves have different slopes, for fast diffusion, the longer the reach, the less potent is the phosphatase. And that's because if membrane diffusion is fast, then you're dealing with a much larger confinement length. And the confinement length defines a bigger volume. And since we're dividing the rate constant by the volumetric rate constant by this confinement length, the bigger the confinement length in the limit of fast membrane diffusion, the slower the reaction. So you have these two different domains. And it turns out that if you look at these numbers, and they didn't really make a big fuss about this in the paper because they were really interested in this reach idea, but it turns out that the actual diffusion coefficients for the case where you have a sort of a reach limitation where the diffusion is slow enough so that the longer the reach, the better the reaction. That turns out to be a very slow diffusion coefficient, which basically doesn't pertain in most real situations. So the real situations really correspond to these situations where we're talking about confinement length as the key parameter rather than reach. Okay, I hope everybody understood that, but we'll reinforce it soon. Okay. So let's go back to the overall problem. How do we determine 2D rate constants for cell scale models? So we're assuming that we can write down this mass action rate equation, the change in the concentration of our product with respect to time is equal to the rate constant times the concentration of the product of the reactants. Okay, so you have A plus B going to C to AB, and this is the simple rate equation for that reaction, right? You all remember that from freshman chemistry. And the question is, can you use the same rate constant for this situation versus this situation? And the answer is obviously no, because this situation, this situation here is membrane tethered, and this situation is free in the cytosol. And if nothing else, they're completely different units, right? The units for a membrane reaction is micrometer squared per molecule per second. The units for a volumetric reaction is per micromolar per second. And as I said, one way to simply convert those is to divide the volumetric rate constant by this confinement length, the length of the tether. And that gives you units of, that have units of per area per quantity per second. And the simple way to make that conversion is shown here. The volumetric rate constant in those units of per micromolar per second divided by a proportionality constant that gets you from moles per liter to molecules per micrometer squared, and then H, the confinement length. So you can do that simple conversion, and maybe that'll work. The question is, how do you know what the confinement length is? That's question number one. And question number two is, how do you know when this system, this all depends on this being reaction rate limited. How do you know when the system is really reaction rate limited and not diffusion limited? So that's where modeling can come in and help. So again, we talked about spring salad. I'm not going to go over it again, but we think that spring salad can help with this. How can it help? Well, you can do a simulation of this kind of system. Here we have just a simple dimerization reaction, two binding sites tethered to the membrane. You can see them here. These red binding sites are tethered to the membrane with a gray spherical anchor in the spring salad software. And when they get close enough to each other within that reaction radius, there's a probability of binding. Okay, so here's what the simulation looks like. I'm going to show it to you again, so I'm not going to go through the whole thing. Let's just stop it at some point. And you can see that up here, there's a pair of bound, and there's a pair of bound. There are a few, and here's one that's bound. And then there's still a lot left that are unbound. We're going to go back to that simulation in a little bit. So you can do that. You can do that simulation in spring salad. But importantly, spring salad uses volumetric rate constants as inputs, because as far as spring salad is concerned, it doesn't matter that those binding sites are tethered or free in solution. You can use the same volumetric binding site that you can measure using in vitro assays. And so everybody, that's the key concept here, is that it just happens to be that those binding sites are tethered by a 5 nanometer tether to the membrane. Doesn't really, as far as spring salad is concerned, it doesn't really matter. We can still use that same binding constant. So now I can do a simple comparison between, let's say we have 50 molecules on a 250 by 200 by 200 nanometer membrane versus 50 molecules in the cytosol, or in a compartment that's got that same XY dimension, 250 by 200, but the Z dimension is now a volume of 100 nanometers. And we're going to, for the membrane tethered molecule, we're going to use a 5 nanometer linker, which is a little bit short for many membrane anchored binding sites, but it's still sort of reasonable. So certainly for lipid bound proteins, lipid anchored proteins. So for lipid anchored proteins, this is very much what you would expect. And here's the difference. The actual rate, we're not measuring a rate constant yet, but the actual rate using exactly the same on rate for the membrane dimer versus the volume dimer is much faster. So membrane dimers come together and go to completion much more quickly than volume dimers when the volume is 100 nanometers. And that makes sense because we're taking the rate constant. If you think about it in terms of the membrane effect, of the confinement effect, we're taking the volume that we've used in the volume dimer case and contracting it to just 5 nanometers. So instead of having 250 by 200 by 100, for the membrane tethered case, you can think about that as being 250 by 200 by 5 nanometers. So it's a much more confined space. There's a much higher local concentration of the binding sites within that small volume, and therefore you have a much faster rate. Okay? Hopefully everybody follows that simple argument. Okay, so that's the idea. And we decided to pursue that idea and use it as a way to actually estimate 2D rate constants for real situations. And the basic approach is to, let's say we're once again doing this dimerization of two tethered molecules. We'll do 100 dimerization simulations so we can get a lot of statistics and try to fit those stochastic simulations to a 2D on rate, a 2D differential equation. And this is what that process looks like. We take this volumetric rate, which in this case was 0.47 per micromolar per second. We let the simulations run in Spring Solid, do 100 of them. We had more than just 2 molecules. We had, I think, 50 molecules in these. I'm just showing you 2 here. So we have 50 molecules and 100 simulations, and we take the average trajectory, that's in orange, and we now fit it to a 2D rate constant based on the surface density instead of the volumetric concentration. We're now using a surface density, and we can fit a rate constant. In this case, it happens to be 0.109 micrometer squared per molecule per second. So that's a 2D rate constant that fits beautifully. As you can see, the fit is shown in black here. Beautiful fit, nice mass action kinetics in 2D. Okay, so that's our plan. That's the simple idea, the 2-step workflow to estimate 2D reaction rates. Okay, so we can, of course, also do the same thing with just using the confinement length. In Spring Solid, for that particular case, we knew that the confinement length was 5 nanometers. So the question is, how well does just doing that simple back of the envelope calculation, which you can do with a pencil and paper, compare to the fits that we get with Spring Solid? And you can imagine that there are other factors that can contribute to the rate of reaction at a membrane that might not really pertain so much to the rate of reaction of a freely diffusing and tumbling molecule in the cytosol. And so the most important one, of course, is membrane diffusion limitations. As I mentioned, this confinement length idea really depends on the fact that the system is rate limited, that the rate of reaction is slow on the time scale of diffusion in the membrane lateral diffusion. The other thing it can depend on is the surface density. We'll see that. It can depend on steric constraints. What are some of the steric interferences between the binding site and its binding partner. Molecular flexibility. What happens if the molecule isn't completely able to sample the entire volume above the membrane? What if it's rigid? And obviously, binding site proximity to the membrane, that's the key factor that we've been talking about all along. So before we get into a real example, which I'll show you one of, let's probe these ideas using this simple homodimerization reaction that I've already shown you, where two identical particles combine to each other, and where we're confining it to 5 nanometers above the membrane. So this is an anchor over here. This is the binding site. And we're dealing with simple dimerization of these molecules. We're only going to have a single stiff spring that holds the molecule to the membrane, but that binding site can pivot around that anchor. So you can have a sort of like a spherical shell of reaction, of a spherical shell that defines the region of potential reaction between one sphere and another. So it's as if two spherical shells are able to diffuse at each other and away from each other. And when they get close enough to each other, then there's a possibility that the two binding sites can form a dimer. Okay. In all of these cases, the binding site diffusion coefficient is 1 micrometer squared per second. H is 5.5 nanometers, or 0.0055 micrometers. So this distance from the membrane to the tip of the binding sphere is 5.5 nanometers, but the distance from the center of the sphere to the membrane is therefore 5 nanometers, because this is a 1 nanometer diameter sphere. Okay. So that's the baseline model that we looked at. And in this case, we're keeping that model. So it's the same for each of these simulation results, but we're going to vary the on rate, the volumetric on rate. It's either going to be 0.01 or 1. So those two cases. We're going to vary the surface density of the membrane-bound molecules. It's going to be either 2,500 or 25. And we're going to vary the membrane diffusion coefficient. Remember, the volumetric diffusion coefficient is 1. We can also vary the membrane diffusion coefficient, change it from 0.01 to 1. So a membrane diffusion coefficient of 1 is sort of similar to a lipid-bound protein. Membrane diffusion coefficient of 0.01 is sort of typical for a channel or a protein receptor embedded in the membrane, a protein embedded in the membrane. So these are sort of two bracketing extremes of membrane diffusion coefficient. They're both faster than the ones that were used in that paper that talked about reach. So this is, yeah. So we also compare this to the simple equation where we convert the volumetric rate constant, which is sort of our input, our sort of our experimentally determined rate constant. And we convert it to sort of this ideal confinement length-based rate constant by using that formula I told you. So the 2D rate constant is equal to the volumetric rate constant divided by 602.2, which is the conversion constant, and h, the confinement length. Everybody following that? So for all of these cases where we have 0.01, that number turns out to be 3 times 10 to the minus 3. When we have 100 times faster rate constant, that number turns out to be 0.3. Okay, just 100 times more. And what we're really interested in is comparing these sort of idealized rate constants, 2D rate constants, to the ones that we determine by fitting the trajectories, the 100 trajectories in our spring salad simulations to a 2D rate constant using Excel, or you could use Gopasio Virtual Cell as well. You can do it in Excel because there's an analytical solution to it, and you can just fit it to the analytical solution. So these are the fit on rates based on the spring salad trajectories. And you can see that for the case where the on rate, the volumetric on rate is slow, it's not very different from the on rate that you determined just from the simple formula, okay? 2.9, 3.1, 2.4, 3.0, they're all pretty close. The one that's furthest away is the one where the membrane diffusion is slow, and you're only dealing with 25 molecules, okay? Not 25 molecules, but a surface density of 25 molecules per square micrometer. The actual number of molecules in all these simulations is the same. We just changed the surface area of the membrane to change the surface density. So this one is a little bit of an outlier, and it seems like there's some deviation from this simple equation. But look what happens when you increase the on rate, especially for cases where you've got slow diffusion. Now there's a much bigger difference between the sort of idealized rate constant and the spring salad fit. So for these cases, are reaction limited. The diffusion coefficient is greater than the rate, and therefore there's a nice match between the simple formula and the spring salad determined fit. But for these cases where diffusion is, where the rate is faster, these are, we call them diffusion influenced. The diffusion rate is about the same as R, and so you really have to take the rate constant from this fit if you want to create a good model. And then at the bottom here, we have two cases where these are all irreversible. In these two cases, we have reversible situations. And we only looked at the case which would be the most diffusion influenced. And we see that we get somewhat closer correspondence to the idealized rate constants when we have reversible rates. And I think I can explain why that would be true in a minute. So I told you that we're fitting all of these to simple analytical expressions, mass action analytical expressions for the surface mass action kinetics. And the fits are nice for most cases. So this is a case for a slow on rate and also a slow membrane diffusion. The fit is pretty good. Here, the membrane diffusion is faster. And the volumetric on rate is faster, and it's not quite as good, but it's pretty good. And here's a case where you've got slow membrane diffusion. We're approaching diffusion limiting and a fast on rate. And here the fit is not so good. This is the orange is the fit, and the blue is the spring salad simulations. And it's not a factor of our not doing a good parameter estimation. It's a fact that the model, which is a mass action rate equation, does not really work as well for this situation where it's starting to have some influence from membrane diffusion. So it's not so bad that you can't use this, especially if you've got high densities of molecules, but it's something that you need to worry about if you're actually doing these kinds of simulations. So the case with this last case where the anchor diffusion coefficient was low and the on rate was fast, was a density of 2,500, does not seem to fit simple mass action kinetics. And again, this is not a new idea, but it's well known that that can be the case for membrane reactions. But it's nice to be able to quantify it and to detect it in this kind of sort of calibration of the kinetics. So let's look at that particular example in more detail. Well, let's look at one of these trajectories. This is the same movie that I was showing you before, but we're now going to look at it and especially look at the final frame of the movie. So these things are starting to interact with each other. Again, this is a case where we're dealing with diffusion. I would call it diffusion influenced. The fit to mass action isn't really perfect. And that's because, as you can see, these individual anchor points are really not moving very far in the membrane. The diffusion is slow over this time scale. And some of the molecules that were originally close enough were able to find each other. Here we are at the end of the simulation. Some of the molecules were able to find each other and form bonds. You can see them all here. But then there are some that, are just too far apart. And you can imagine that as the simulation proceeds, those isolated molecules are never going to be able to find a partner. This guy here is probably never going to be able to find another partner. This guy here, maybe he'll find this guy eventually, but it's going to take much longer than when two of those guys were close to each other. And that's why this can never really match mass action, because mass action doesn't care about diffusion. Mass action is completely ignorant of diffusion and is always in a limit where diffusion doesn't matter. And so that's why we have that kind of situation. Here's an extreme case. Here we're taking the diffusion of the anchor down to 10 to the minus 6 micrometers squared per second. And as you can see, there are a few guys that were probably close enough to each other at the beginning that they were within reach, and they found each other, but they're never going to be able to find all the rest of them. And here's the attempt to fit this to a mass action rate expression. Okay. So we can use this kind of simulation to tell us when we're within a proper regime that we can use mass action kinetics, or at least be aware of the fact that we're not able to perfectly use it. Let's think about some of the other issues. So here we're doing some other simulations where we're trying some other of the ideas that I mentioned earlier. So first of all, we're just increasing the linker length. All of these cases now have a 20 nanometer distance between the binding site and the membrane, but different other scenarios. So here, this one here can be compared directly to the previous case. You can think about this as being essentially 4 times longer than the 5 nanometer case. And indeed, if you just apply this formula instead of 0.3, the on rate that you calculate from just the confinement length is a factor of 4 lower than what you had before. And here are the spring solid fits. Because this is a lower on rate, even though we're dealing with relatively slow membrane diffusion, these are all pretty good fits to the K-on that we calculate from the idealized formula. So when you're dealing with a longer system where the on rate, due to the higher volume here, the lower effective concentration of the binding site, you have a much lower on rate, and therefore you're back in this domain where your mass action kinetics are pretty good. Oops. The other thing here is that some of these other factors don't matter too much. Here, this is including some steric sites to make it harder for these molecules to hit each other. And here we're including some flexibility. Flexibility actually increases the rate a tiny bit. The steric sites compared to the just the stiff steric sites. The steric sites, on the other hand, decrease the rate as you would imagine, but it's not by very much. So what we can conclude from here is that that this confinement length is the primary factor in determining these on rates. But how do you determine that confinement length when you're dealing with a much more complicated molecule? So let's talk about, in the final few minutes, let's talk about a real world case. In this real world example of using this idea of determining 2D reaction rates from spring solid, let's look at this particular example. Here we have a very well-studied system, the activation of the RAS GTPase. And the RAS GTPase is activated by a GTP exchange factor. In this case, we're using a particular one called SOS that binds to RAS at two different sites. SOS binds at an allosteric site and at a catalytic site. It first has to bind the allosteric site before the catalytic site is active, and then the catalytic site can cause the exchange of GTP for GDP on RAS. Now, all of this happens at the membrane. RAS is a lipid-anchored protein. The SOS is initially in the cytosol. It gets recruited to the membrane by an adapter protein called GREB2, and GREB2 is recruited to the membrane because it has an SH2 domain that binds to a phosphorylated tyrosine. This mechanism is applicable to EGF receptors. In this particular cartoon, it's the LAT receptor in immune cells and T cells, T lymphocytes. And here's, so this, all of these molecules, the EGFR, the GREB2, the SOS, and the RAS have PDB structures. In fact, I don't think SOS had a PDB structure, but we used AlphaFold to approximate the SOS structure. Probably not so much an approximation. It's so good. But we got structures for all of these, and we created this system. And here's sort of our spring salad model developed on that basis. This is just showing a dimer of EGF receptors. Only one of them in this case did we bother to put the phosphotyrosine linker on. So here's a phosphotyrosine that's bound to GREB2. This is GREB2, and here's SOS. And SOS binds to an SH3 domain on GREB2 through a proline-rich motif. And this part of the SOS molecule is the action part. I think this one is the allosteric site, and this is the catalytic site. Okay. So we're looking at two scenarios here, and how this system works and why the allosteric site is required is a major focus of a lot of research. And I'm not going to go into that research here. I'm just going to show you a very simple result from these kinds of spring salad fits. So here's just a simple binding. Obviously, before the catalytic site can cause the exchange of GDP with GTP, it has to bind to RAS. So the SOS catalytic site has to bind, has to bind to RAS. So here's the catalytic site here, and here's RAS on a membrane. And we're just, those are the two reactants, and we're just gonna allow them to react, and we get and we know what the volumetric rate constants are because those have been measured. This is it here, 0.27 micromolar per second, per micromolar per second. And we know what the K off is also. So those are all things that have been measured. And we can do a spring salad simulation, 100 trajectories, fit it nicely to 2D reaction kinetics, and get an on rate in 2D units of 0.0011 micrometers square per molecule per second. When we do the same thing, but first have the SOS molecule bind to RAS at its allosteric site, and then ask how fast does the SOS catalytic site bind to a second RAS, we get a much faster on rate. And you by now you can intuit why that is. It's simply because whereas this system was flopping around in the wind here and has a confinement length that's very high, here we've condensed the confinement length by having this first RAS molecule bind to the allosteric site, and therefore this catalytic site is constrained to be much closer to the membrane and much closer to the RAS molecules that are floating around in the membrane to bind to it. So this reaction is about 4 times faster and has a faster on rate. And this is how you can estimate those on rates based on spring salad simulations. Okay. So to conclude, we developed a computational method for converting experimental rate constants from 3D to 2D that accounts for factors unique to membrane binding reactions, diffusion of membrane anchors, the unique geometry of the confined binding site, any other steric interactions, etc. But the site distance from the membrane seems to be the strongest predictor of the reaction rate constant. And you can, if you have no other recourse, you can certainly produce an estimate by using by trying to estimate that confinement length. So the important point here is that, doing these spring salad simulations are very practical, but they're not practical for a big signaling system. What this now allows you to do is to find these parameters that you can use with some degree of confidence in much bigger models that you might want to run in COPACI or VCell that require those membrane rate constants, those surface-based rate constants. So with that, I'd like to acknowledge Kelvin Peterson, who's been in my lab for a number of years now, at least on a part-time basis. He started as an undergraduate and is now a medical student here at UConn, but he continues to work in my lab. Spring salad was developed, as I mentioned earlier, by Paul Michalski, and Ani Shadaraj actually continues to give us some support in our work, as well as Dan Vasilescu, who's porting it over to VCell. There's the VCell team and the COPACI team, because when you get to more complex kinetics and want to do this kind of fitting, using the COPACI parameter estimation turns out to be very useful. And those are the NIH grants, and all of this work was done at our Center for Cell Analysis and Modeling at UConn. So thank you.
Thank you, Les. Are there any questions? Because I have a question. Les, if you can go one slide down, back. Oh, one more slide. Yeah, here. So this is initial configuration of SOS that has this hook, right?
Yeah, I mean, that's just meant to so that you can see this in the same way that you see this. I mean, in fact, this all of these sites here, these yellow sites, all of them are very flexible. So this thing is flopping around in the cytosol in a much more extended conformation in general. Compared to this, where the binding of the pre-binding to the allosteric site is really holding the catalytic site much closer to the membrane.
So this screenshot is kind of the first millisecond of simulation, right? Because after that, it probably will flop on a closer
Right.
to the membrane.
Yeah, exactly. It's probably time zero of the simulation, not the first millisecond.
Mm-hmm. Yeah, thank you. So any